Here to welcome the guests on the panel is the moderator, award-winning biotechnology journalist Luke Timmerman. Luke specializes in life sciences. He's the publisher of the Timmerman Report and has served as national biotechnology editor for Xconomy and national biotechnology reporter for Bloomberg News. He got his start covering life sciences at the Seattle Times. Please welcome Luke Timmerman.
All right, thanks a lot, Ware. It's a pleasure to be here tonight. What a great panel! I think you're gonna find this to be a real treat, learning from these folks about emerging technologies in health. I think panelists, you're all welcome. Come right up, and I'll introduce you as you arrive. But I see Atul moving here. Okay. So first up is Atul Butte. He's going to sit right here to my left. Yeah. And Atul is, he's running an institute at UCSF. He moved there from Stanford about a year ago, working on computational health sciences. So he's putting together a massive database, trying to put sense to bring some sense to it out of all the people in the UC system. It's a pretty amazing task, and I'm eager to hear him talk about that some more. Right to his left, Carol Dahl. She's the executive director of the Lemelson Foundation in Portland. It fosters invention. So they educate people about the role of invention in society. An underappreciated one. And before that, that job, she was at the Gates Foundation here in Seattle and spearheaded work on the Grand Challenges program there, which again fosters really inventive, kind of off-the-wall kind of ideas for global health to shake things up. To her left, Jacob Korn. He's at the IGI, and he can tell us more about that. It's a relatively new institute at UC Berkeley. It's focused on genome editing. So those of you in the audience who've heard about CRISPR, he's your guy. And to his left, Lee Hood. He's the founder and president of the Institute for Systems Biology here in Seattle. Obviously, very well-known scientific entrepreneur. I happen to be writing a book about this guy. Maybe when I finally get it done, I could have an event here at Town Hall, and you're all welcome to come out and hear more about that. And on the end, we've got Roger Perlmutter. He's the president of Merck Research Laboratories, executive vice president of Merck and Company, one of the most important jobs in the pharmaceutical industry. It's one of the most robust drug discovery enterprises in the world. He has long-time Seattle ties. He has ties to that guy sitting next to him, Lee Hood, who he was a graduate student, or actually a postdoc in Hood's lab way back when at Caltech. And I don't want to give away too much about the book, but one little morsel. Roger joined the faculty at the University of Washington as a young immunologist and played a key role as a behind-the-scenes broker in recruiting Hood from Caltech to the University of Washington. And so a whole lot of things that spun out of the Lee Hood orbit later in Seattle, you can sort of trace that back to Roger Perlmutter. And any disagreement there, Roger? He's being modest. Okay, so amazing things happening in the world today in biotech. I mean, just prepare for this event tonight. You can think about so many things that you've probably heard about in the news. CRISPR, genome editing, science's breakthrough of the year. This idea that you can edit the genome of multiple species quickly, cheaply, easily. Any graduate student can learn it. It's both thrilling and a little scary at times. But there's amazing things going on in gene therapy, RNA therapy, organs on a chip. There was an interesting talk about that today at Hood's Institute. So bioinformatics, like the software that it takes to make sense of electronic medical records and connect that to what's going on at an individual genome level. Fascinating stuff. So before we dive into some of the questions that I have, I'd like each of these folks to say a little bit more in their own words about what they're doing. What excites you about what you're doing these days? You want to start, Atul?
All right. So, do you want me to go first?
Yeah.
All right. So, thanks for coming out tonight. My name is Atul Butte over at University of California, San Francisco. It's a real pleasure to be here, and I think it's really important for the public to learn what we're doing and not always see the scary side of what we do, but the most amazing potential. This is the most amazing time to be in research. So I'm privileged to be at UCSF. I was actually at Stanford for 10 years. I just moved just a year ago to UCSF. I moved there to think about what we're going to do with all of our patients. We're collecting a lot of data on patients. You're going to hear that from all of us, I think. Right now we're in this era called precision medicine. A lot of people have different definitions for this. We want to customize the medical care we deliver to you, but also take into account what we've been doing to everyone else so we can learn from everyone else. So it's not just about you, it's about everyone, right? And the idea is that it's really the collision of two amazing fields that we happen to be spending billions of dollars on. One is this field of electronic medical records, where every hospital around us wants to capture electronically what we want to do to patients and what we are actually studying, what we're actually getting from patients. That's 100 billion that way. And all these molecular measurements in the genome, and these two coming together, and to me, that makes precision medicine. So I get to run a new institute at UCSF to try to recruit faculty, to come up with inventions, spin some out as companies, as I'm sure you're going to hear about over the next hour or so.
Great. Carol?
Great. Yeah, so as was mentioned, I'm from the Lemelson Foundation. It's a private family foundation that's based in Portland, Oregon, but our founder was one of the most prolific independent U.S. inventors. He had over 600 patents to his name, believed in the power of invention, and started the foundation because he believed that the future was about invention and that the history of the United States and our success, our quality of life, our strong economy was based on that. So we work on improving lives through invention. We work in the United States around that goal of strengthening our own way of life here, but also in developing countries. We look to solve the problems for those who are the poorest, the most vulnerable populations in the world. And we're excited about science and technology and what it can mean in terms of underpinning invention being ideas that can be put into application and have impact. And so we do that through inspiring and educating a next generation of inventors and then helping to create an ecosystem where they can take their ideas and turn them into products and impact. And I would say I'm really most excited on two fronts. One is that we see, particularly here in the US in our programs, the power of students and the graduate students and postdocs coming out of the universities to be the engine of change for a better tomorrow. There are huge challenges that the world faces right now, and those students are well equipped. In fact, I bet everybody sitting here has had students coming out of their labs that are going off to start companies with really valuable, socially impactful products that are going to change the way we live for the better. In the developing countries, what we see is the power of science and technology through invention actually to make lives better. So we're seeing excitement about new tools and technologies that are really being developed, not as stripped down versions of what we use here, but really co-created with the local populations to solve their problems in ways that can reach and change even the lives of the most vulnerable people. So those are my two areas of excitement.
Great. Thanks, Carol. And Jacob has experience with applying academic ideas in industry as well, right?
Yeah, so I'm currently the scientific director of something called the Innovative Genomics Institute, and before that, I was a group leader at a company called Genentech down in the Bay Area. I still remember pretty clearly when some of the first CRISPR papers came around. People started talking about it as a gene editing thing. I mean, a chill just went down my spine because it was clear that basically everything was different now. Gene editing is not a new thing. The technology to do it has been around since the 90s, but it's been very slow, very difficult to do, and really the power of CRISPR gene editing is that it democratizes it. It makes it as fast and as cheap and as easy as our ability to read genomes. So now, finally, our ability to change genomes has caught up with our ability to read genomes. And I think that has huge implications for treating genetic diseases and creating cures for genetic diseases, but also has a lot of implications for agriculture, for knowing how we're going to combat climate change, what we're going to do agriculturally, environmentally, it also has a lot of implications for just basic drug discovery. One of the things that we ran into all the time in drug discovery at Genentech, and I think at any company, is just how do you make models of the diseases that you're working with, and that can be extremely laborious and extremely difficult. And suddenly, it's the type of thing that you might do in a week, as opposed to do over the course of 4 years, something like that. So it's really gene editing is not just about genetic disease. I think it's one of those things that's going to have huge, far-ranging implications for basic discovery, for understanding of the world around us. And there's some interesting stories around that, which I'd be happy to share with you guys if you're interested.
So Jacob, maybe this is totally out of date, but I remember when George Church published the paper that they edited something like 60 genes in a pig simultaneously.
Yeah, so that was, it was a little cheating because it was the same sequence. It's just repeated several times. So there's a virus that's present a lot of times in the genome, and so they hit that one gene, and then, of course, they hit that sequence, and so it edits ... all those times. It's not to trivialize it, right? It's a very difficult thing to do because that sequence they're trying to go for is a viral sequence that infects that pig. And so they're trying to remove a virus from the pig to use it as sort of a medical research tool.
Okay, but the idea is that you can...
Small step for pig.
Exactly.
Well, I think the point is that you can edit multiple genes, not just one gene.
Yeah,
right?
that's absolutely right. That's absolutely right.
Okay, next up, Lee. And I sort of forgot to mention that, Lee's claim to fame is that he led the team at Caltech that developed the first automated DNA sequencer, which is really, the epitome of an enabling technology. It enables so much of what goes on in everybody's lab today. The genome is now something like $1,500. Is that the latest? Roughly what it costs?
Yeah, and hopefully within a year and a half it'll be 300.
Yeah, so the things that people can think about new things and do all kinds of experiments they couldn't. That's enabling. So Lee, what excites you today?
So what excites me today is I think we're at an enormous tipping point in healthcare that is going to absolutely transform how we take care of people. One aspect of that is the pioneering development of systems medicine, which takes a global holistic view at attacking very complicated problems. A second really key technical strategy is that idea that the essence of systems medicine for the future will be for each of you that will develop a dynamic, dense, personalized data cloud of billions of data points, and will have the ability to analyze that and identify for each individual the actionable possibilities that will let them optimize wellness and or avoid disease. And these dense dynamic data clouds are not only the key to optimizing human potential through the optimization of your wellness, they're the key to understanding how we transition from wellness into disease, how we identify the earliest stages and use systems approaches to get the diagnostics and therapeutics that can reverse that disease before there are any symptoms and any irreversibility. This will be the preventive medicine of the future. And the final point I would make is that on March 9th, ISB had an affiliation with Providence Healthcare System that is going to enable us to bring this vision of healthcare to the bedside. And that is unbelievably exciting for me, something I've been working for a lifetime to be able to do.
Great. So last but not least, Roger, you've got one of the important new immunotherapy drugs for cancer on the market. Happened to put a former president into remission. That was a pretty exciting thing. Can you tell us a little bit about that?
Right. So when Lee's dynamic, dense, data-rich clouds don't work for you, then then
You don't deserve it.
then you got to deal with us. And you know, my organization, Merck Research Laboratories, is the premier, has always been the premier research-intensive biopharmaceutical organization devoted to the development of medicines and vaccines to improve lives around the world. And we have, over the last several years, been able to translate a vision which is more than 100 years old. The idea that one could harness your own immune system to actually tackle the challenge of malignant disease, of cancer. It's something that many of us have dreamed about. Certainly we thought about a lot when I chaired the Department of Immunology at the University of Washington. But it has now been realized in a kind of spectacular way. And the example that everyone knows is, of course, for former President Jimmy Carter, who was diagnosed with widely metastatic malignant melanoma with lesions in his brain. For someone in that circumstance, typically the survivability in that situation is very low, and the time to death, unfortunately, is very short. But with the availability of new immunotherapies, remarkably, immune system revs up, recognizes the tumor wherever it might be, and actually either eliminates it or suppresses it to such an extent that life continues. And as you can see from, well, I guess you can't exactly see, but as you hear about from Jimmy Carter's, weekly Sunday school sermons, doing pretty well. And that's a remarkable finding, and it opens up enormous horizons. So we have been examining now the entire spectrum of cancer in human populations, and what we found is that by blocking a single signal on the surface of immune cells, we can reveal the pre-existing immunity that exists in a large percentage of cancer patients and see exactly this kind of stabilization, and it appears to be durable in many, many cases and has relatively few, it's not free of side effects, but it has relatively few compared to traditional toxic chemotherapy. It's frankly the most exciting thing that's happened in cancer treatment in more than a half century. And so we like coming to work in the morning. It's pretty good.
Now, it doesn't work for everybody, to be fair.
Yeah.
And but part of what we're learning about immunotherapy is it turns a lot of biology, on its head, or a lot of conventional wisdom. So in the case of Jimmy Carter, when your prognosis at age 91 with a situation like he describes, it's really been pretty grim historically. But with a drug like Merck's, and there are a couple of others that work through similar ways, it works by releasing the brakes, so to speak, on the immune system, or a cloaking mechanism on the immune system. And when you pull that back with someone who's 91 years old, they've naturally accumulated a lot of mutations because you've had a lot of time for your cells to divide and develop, errors. And those mutations are what the immune system then recognizes. It says, whoa, there's a lot of things abnormal about this thing. And so he actually had, because of his age, we think, had an even better prognosis with a drug like Keytruda.
I'm
Maybe.
being a little careful. I mean, I think, first of all, I think we were quite surprised that a 91-year-old, we weren't that surprised. I mean, he's a pretty vigorous 91-year-old, but the immune system does undergo a senescence process, and we know that because we make a lot of vaccines, and we know when we vaccinate elderly people that the vaccination frequently doesn't take for a variety of reasons. So the immune system is less good. And also, while you're exactly right, Luke, that there is a correlation between the number of mutations that are present in a tumor, and all tumor cells have quite a few mutations, and many tumors have sustained defects that make them more prone to accumulate mutations. That those mutations are correlated with the response to these new therapies. We don't know for sure that the mechanism whereby the tumors are dispatched is because of recognition of those mutations. We think that's probably true, but there are other potential explanations, and we're still just at the beginning of understanding why this works so well in those people in whom it works.
Jacob, maybe you could talk a little bit about CRISPR, and I know you alluded to why this is so exciting, but what does anything worry you about it? There's been a lot of talk about that in the public sphere.
Well, I think the thing that you'll see on the cover of almost every magazine, news article, whatever, the two words that I get asked by everyone is designer babies, right? It's always designer babies, designer babies. And I think that's a little misplaced. I mean, I think maybe that's something that we would want to be thinking about in 50 years, 100 years. But a lot of these things, we don't understand what makes, one person a super genius and another person, a normal mortal like those of us, we don't understand what makes, someone an incredible sprinter and another person just, pretty fast. So these are really complex traits. But there are 6,000 genetic diseases where they're monogenic, and we know exactly what the mutation is that causes them. And so I think, to me, and for those of us working in gene editing, that's kind of the low-hanging fruit that we're very excited about, right? If someone were to want to make a designer baby, we wouldn't really know where to start. But if we want to start curing people with genetic disease, we know exactly where to start. We know exactly what to do.
For the single gene diseases, the more complex multifactorial ones like diabetes or schizophrenia.
Exactly, autism, things like that.
Yeah.
I completely agree. I mean, this is maybe where, Atul can come up with markers for, what's actually causing some of these things. And
Well, I think there are a lot of patients out there. I mean, so every day, every week, every month, major medical systems, pediatric hospitals are discovering new disorders, new syndromes. This really goes back to 2011. I think the first of these kind of kids came out of Milwaukee, where there was a kid that was just going to die. Spent the first 5 years of his life in the hospital, in pediatric hospital in Milwaukee, and just kept killing off his own colon, his own bowels. And kind of Hail Mary pass, let's sequence the kid, sequence the parents. They found a gene that suggested to do a stem cell transplant in the kid, and he's fine. He's not out of the woods, but he goes to school, he eats a regular kind of diet, and that's just in 2011. It's 2016. That was 5 years, and every children's hospital now gets these kids, and parents, especially the parents that are frustrated with how long it takes to figure out a diagnosis. What we call these diagnostic odysseys. And one application of a genome sequence on the kid and the parents, of course, there's a lot involved, can really get to a syndrome. Now we have a long way to go to figure out what to do with these kids. They don't always have something clear cut to do. But we can start to subdivide these diseases. I mean, you can imagine a disease with just one kid in it, because you don't even need a second one yet, because you got enough to define it with the first. So, technologies like CRISPR-Cas, I think, are a hope that maybe we could offer something to all of these kids. But we're going to get a lot more of these kids and a lot more of these diseases before we can do something about them. That's kind of the depressing part of this whole genome revolution, I think.
Mm-hmm. We're in an information gathering phase, I think of it. I mean, do you know, Atul, how many human genomes have been sequenced, what the latest count is?
Yeah, people ask me. I'm sure Lee knows this number, so he better, whatever number I give you, he's probably going to correct me. But I'm guessing probably complete genome sequence with what's called the 1,000 Genomes Project. I'm guessing probably 30,000 or so, maybe. No, let's see. Entire genomes. So just a subtlety for the aficionados in the audience, you get your entire genome. So what's the easiest way to think your genome? I'm sure Lee has an analogy. For me, it's the joy of cooking. How many of you have the joy of cooking at home? It's a definition of wishful thinking, right? You buy it, but you never cook from it, right? It just cooks itself. So I don't want to use the Bible or the Quran, but I use the joy of cooking because that's a neutral book. So you know how thick that is. And so 2,000 copies of that are the number of letters in your genomes. That's about 6 billion. 3 billion for your mother, 3 billion for your father. That's 2 statues of liberties tall. And so 1 in 1,000 of those letters are different between us. So that's what your genome is. And I'm guessing probably, so you can get all of them, or you could just get the recipes. Maybe the important part of the recipes, that's the coding region, what we call the exome. Or do you want the table of contents? Do you want everything else? You probably want everything else if you can afford it. So there's a difference. So do you want the 1%? Do you want 100%? It depends on what you can spend. But if you talk about the 1%, the exomes probably like, I don't know, probably 500,000, I'm guessing. If
Yeah.
you want the whole thing, probably like 30, 40,000, I'm guessing.
Yeah, but the 23andMe test, a lot of people have probably heard of. That's $150, and
Yeah.
that'll give you that table. of
Couple letters
contents.
each page,
Yeah.
okay? But
Yeah.
they're the right letters, okay? They're not the wrong ones. They pick some good ones. They don't
Uh-huh.
pick useless ones. They pick some good ones. And from those letters, you can kind of tell what the letters are around there, if you can imagine what I'm stretching this analogy now. So they're not completely useless either, but it's also cheaper.
But I mean, 15 years ago, we had one genome.
Yeah, yeah, or a
Human
collection
Genome
of
Project.
30, let's say, that were mixed up randomly, let's say, right? But
It was one composite genome, and this is just an information explosion. And now the task is figuring out what it means and what to do with it. Very interesting. Carol, you are focused on global health applications. Can you tell us a little bit about where does this genomic stuff have application there, or are we getting too far ahead of ourselves?
Well, I think, first of all, just to clarify, we work on things in global development. So it's not just health. We also work on agriculture and actually whatever the problem is that needs to be solved. And so it'll kind of relate to my answer, which is that we probably are way far away from the idea of precision or personalized medicine in developing countries just because the basic needs are not being met. People don't have access to clean water. 40% of the world, like 2.6 billion people, don't have access to sanitation. So toilets are needed that can work in these environments. So there's some major barriers to be addressed first. But I think genomics and maybe the derivatives of that, whether it's proteomics or transcriptomics or so on, are all going to be essential in terms of understanding some of the fundamental problems we face. We talked a little bit today in our sessions about preterm birth, which is a problem that is pervasive globally, but it has 95% of the impact in terms of death happens in the developing countries. And so in that case, we don't fundamentally understand why preterm birth is occurring. So I think all of those tools, the tools of systems biology that will help us to understand the underlying mechanisms and causes for births that take place before a full gestational period, that's going to be important. But we also see some of the more sophisticated and cutting-edge technologies maybe coming in places around agriculture, around vector control of transmission, control of the vectors themselves that transmit diseases.
You mean mosquitoes?
Mosquitoes, yeah, but particularly with mosquitoes, we're seeing some of the more sophisticated technologies, even CRISPR, being brought to bear. And so those are places where I think there may be more kind of flashy, high-tech innovations happening at the cutting edge. But there are still fundamental changes that need to be solved in terms of the human health there that start all the way from toilets, but run all the way to fundamentally understanding the biology behind things like preterm birth, the interaction we have with various infectious diseases, why there's an increased frequency and predisposition for chronic diseases in people who have been undernourished in their early years. There's a lot of biology left to unpack where we're going to need solutions. So,
And Jacob, I've heard a lot of people say that CRISPR may see its first applications in the ag arena. Is that something that, do you believe in that, or if so, why?
Well, I think that sort of gets into this whole complex regulatory landscape. Right?
Yeah.
The regulatory landscape around human health and ag is very different. I mean, think about if you want to do some editing in corn or in rice. I think that introducing that, doing those kinds of edits, just in a lab is very different than saying, I want to try editing a person, disease in a lab, right? And I think very rightly so. So, will we see some of the first advances in agricultural crops? People have already started reporting editing in agricultural crops, and no one has yet to even report in a lab setting editing of a person.
Right. We have a company that just went public and is worth $1 billion based on the prospect of genome editing, but it has not yet begun a clinical trial.
Yep.
There are whole other fields of science that are being opened up, and this because of some of the enabling technologies. And I think of DNA sequencing particularly. The microbiome is one area. And I don't know if you talked about that today. I know you have, Lee, in past symposia at the ISB. But I don't know how familiar folks here are with the microbiome, but these are the microbial communities that live in our guts and are part of really who we are. We're learning tremendous amounts because of the sequencing of stool samples. That's part of what you do there at Aravale. That gives you that vista, if you say, into your health. The bugs have a role in how we fend off infections, how we digest food. What are some of the big learnings in that area the last couple years, and where do you see that going in the next couple?
Well, just to put it in perspective, the microbes in your gut are 10 times as common as all the cells in your body. About 30% of the small molecules in your blood come from those microbes, and they have enormous influence and control on your health. It's clear they have an influence on obesity and many other aspects. The days are in the earliest stages of discovery for the microbiome, so we have to be very, very careful about overplaying things. But, there are absolutely spectacular experiments, like you take a human that's fat and his identical twin that's thin, and you transport their gut microbiomes to appropriately prepared mice, and the fat person makes a fat mouse, and the thin person, the mouse remains thin. So that's the kind of impact and influence we're going to come to understand about your microbiome. The technology that's really made this possible has been DNA sequencing of several different sorts. But I think we're at the beginning stages of therapies which will say to you, you're 20 pounds overweight, here's a probiotic, you drink this for the next 3 months, and we'll guarantee you'll lose 25 pounds. Those are the kind of things we can hope for. And I'm not saying we know how to do it, but I do think they have an enormous impact on us. There are critical chemicals we use in our body that are only synthesized by your gut microbiome. So it's an essential, it's the greater ecology of what you really are, who you really are.
Yeah, I wonder, again, with global health, I mean, this is Seattle, we've learned a lot about C-section babies are one great example, right? Where they're not being exposed to the vaginal microbiome upon delivery. And there's now more and more research that says maybe some of these kids aren't getting the same kind of, or they're maybe a bit immune deficient. Or maybe there's a way to compensate for that somehow.
Really hard study to do.
Well, you have natural childbirth, and then you have C-section babies.
Yeah.
You can take vaginal swabs from the C-section mom.
No, the problem is that the measurement of lifetime health as a readout for how well did you do as a C-section versus not. Population-based studies are very difficult to do because there's so much heterogeneity in the human population in terms of overall wellness. I think an insight which Lee, you could speak to and others could speak to is very important is we are seeing, as a result of the availability of precision analytical tools that provide so much information about each individual, we are seeing the rise of what we call the N of 1 experiment, the opportunity to look at a single individual and follow them through the course of their life in terms of the whole set of parameters. Microbiome is one example, but a variety of other measurements of metabolites in blood, in urine, that tell us a lot about the way in which the body is functioning and provide a basis for thinking about regularizing function and moving more towards a healthy physiology as opposed to an unhealthy one. I think that's some of what you were thinking about and talking about when you
Right,
spoke
right.
about the.
Yeah, I'll add
Yeah.
just a bit to that. I think what is really interesting about clinical trials is we'll take 30,000 patients, give them a drug or its placebo, and then we'll average the responses, when in fact, every single individual is unique genetically and unique environmentally. And if you think about a complex population like that, ask yourself, what's the appropriate control? There's no way you could really get an appropriate control. So what we are going to do in the future is use the only control that's proper for you, and that's you. We're going to follow longitudinally your dense dynamic data clouds, and we'll track you across your wellness, and we'll get the slightest perturbations in disease and be able to do things. We recently did a wellness study with 108 different individuals, and each of those 108 was a fascinating N of 1 experiment. And we learned an enormous amount from each of those N of 1 experiments that would have been totally blanketed and confused by trying to average people. The only way we can get the statistics that make human geneticists feel comfortable, not with N of 1, but having 10,000 N of 1 patients, and then being able to stratify them based each on their own unique properties for responses that you're interested in, response to a drug, failure to respond. So all of clinical drug discovery, I think, is going to fundamentally change in the future, and that's because you are the only adequate control for your own human potential.
No, it probably does need to be said that this is quite controversial. The whole movement toward evidence.
Only for people who don't think. That was kind of a joke.
Lee's never been the most diplomatic guy. But no, I mean, to back up just a little bit, what Roger was talking about with the averaging and populations, I mean, so for the example of Keytruda, the melanoma drug, you would need to, in today's world, governed by the FDA, you would need to enroll, say, I don't know, 200 patients or 500 patients with a common diagnosis, like stage 3 or stage 4 melanoma. They all look roughly the same. We just kind of lump them together between the ages of, say, 60 and 80 or something like that. And we randomize them. Half get the drug and half get the standard of care or a placebo. And you try to eliminate all the other variables, like whether that person drinks Diet Coke or ate a turkey sandwich for lunch. It's kind of forget about it. And that's how helps you zero in just on what that drug is doing to provide, proof, the medical evidence, really, that the drug's working. But so this is the problem with N of 1. How do you know that you were drinking a lot of Diet Coke one day and then you stopped and your microbiome got better? How do you know that it's because of that, or was it something else?
You drink more Diet Coke.
When you have 10 or 50,000 dense dynamic data clouds, you'll have an enormous amount of data where you can sort out those kind of variables.
In essence,
He's looking at me for a second.
I was looking at you too.
So the challenge, though, and Lee knows this too, is that we'll always be measuring more things than we have people in our studies, right? So it's underdetermined if you know some matrix math or whatever. And so this does make it hard. I mean, so we can overfit these things. We can find this pattern, that pattern. So everything Lee says, we actually kind of need two of them, right? So learn on one of them and test on another, or at least to kind of look at significance. But it is hard. I mean, so I'll give you an example. I mean, like even if you have 20 years worth of data on people, it's still hard to figure out where did they actually get the disease, what caused them to get a disease. I mean, there's some obvious things, right? We have Cheesecake Factory, we have 1,000 channels of TV. If you watch them, you're not going to go exercise. We know some obvious things. But some things are hard. I mean, so one analogy, the computer scientists know, that computers are really good at playing chess. Now they are good at playing Go. I've got to learn Go so I can understand how important that is, but they're good at that. But actually, if a computer program plays chess or plays Go, and loses the game, it is actually impossible for the computer to tell you why it lost. What step did you actually lose the game? And in some ways, it's actually impossible. It's an intractable algorithm right now. And so in some ways, you can think of, even if you have 100 years worth of data on yourself, where did you actually lose is a tough problem. Like, we can't even do that for chess today. We can win the game, but if we lose, we're not really sure why. So there are certain things. We still need algorithms. Still no computer scientists here. Some things are hard, even with the data. So the best we can do is sometimes learn it with one massive cohort and test it on another one, and maybe we can believe it at that point. Maybe we're not sure why, but maybe we can at least believe it.
Very interesting. Now, I know we want questions from the audience, and I can't really see very well. So if folks want to come up and grab one of these on either side, you're welcome to. Or you can raise your hand if you're more comfortable doing that. Yeah.
Here comes a victim.
Well, clearly data is a very critical point here, and we need probably a lot more of it. So Apple has announced Research Kit, Care Kit, things like that. There's probably dozens of other tools. Any sense on this that you care to respond to?
Yeah, so let's have some fun here. So how many people have a wearable, broadly defined watch, a Fitbit? My God. Okay, so 1, 2, 3, 4. Well, let's say about 20 people are a person audience. So it's about, let's say, 5% of this audience. Silicon Valley, it's typically about 10%, 15%. So actually, but I've been in many audiences where it's 0. So that's a great audience here. So I think a lot of people have these. I'm going to answer first, and I'll toss it over to these guys. I think those devices are great. So first, there's the aspect of you using your own measurements. I totally believe what Lee said. I just retweeted it here. You are your best control. I love that kind of idea. I used to laugh at these people. So I'll tell you a little personal moment here. I used to laugh at these people, these quantified selfers who measure everything that comes in and out of your body. Measuring things that come into your body is tractable. Measuring things that come out of your body sounds gross, right? So I used to laugh at these folks until December of 2012. Came back from a trip to Hawaii, and I stepped on a scale, and I was 247 pounds. If you don't believe me, just look at my TED Talks. And I realized when I hit 250, they can measure me in tons, right? That's a quarter of a metric ton. What the hell happened to me? I'm driving everywhere in Silicon Valley. Like, what the hell happened? So I bought everything Fitbit makes. I'm not even commercially connected to them. I'm a big fan. I lost 50 pounds, okay, in about 2.5 years. It's kind of Weight Watchers rules. You bite it, you write it. So I write down everything I've eaten for two years. I have a scale, even my carry-on bag there. There's literally a digital scale. Weigh myself every morning. I know what we tell kids with anorexia not to do that, but I do it. I know. I'm a pediatrician. I know. It's kind of weird. But I weigh myself every morning because I kind of know what to do next, right? So that helps me. I'm not saying it's for everyone, but these devices, oh my God, if you're ready to accept them and kind of use that to gauge your life, I'm a big believer in these wearables. The use of them in research will explode, but they can't detect cancer yet. I mean, they can detect when you're walking upstairs and all. That's great. So they're not for everything. They're kind of connections to DNA now. So there's this connection Apple announced with 23andMe. I'm sure Samsung and others will announce, and Google will announce those kinds of things too. But still, it's a long way to go to run the kind of research studies that Merck runs with these things. I mean, a long way to go. But to give them credit, I think every pharmaceutical company is looking at beyond the pill kinds of studies, trying to figure out how they could be useful for studies. But I think they can help yourself a lot more than maybe the pharmaceutical companies. I don't know. Let me toss it to these guys here.
Well, it's interesting. I went to a quantized self conference about 1.5 years ago and gave a talk. And these are all the people that make all the measurements or are fanatical about it. And the very first question I got is, I've done quantized self for 2.5 years. Where do I go from here?
Yeah.
And that really is a critical
Great
question. And the answer is wellness is much more than your sleep and the number of steps you take and the number of stairs you climb. That is, those are all important dimensions, but you need to bring in the other things if you're to get a holistic view of what wellness is about.
Right.
So that's my point. So any of you who are interested, we did start a company called Aerovale that is all about scientific wellness and putting together all of the different dimensions. But I'll tell you what has turned out to be the really critical part of that company are the coaches that can come to you and explain, A, what the actionable possibilities we derive from the analysis of your data are, and B, how they fit in the context of what your own health plans are.
Yeah.
And those coaches end up being inspirations and they keep track of your promises. We can all make lots and lots of promises. But when your coach sends you an email 2 weeks in after a promise and says, well, Lee, how are you doing?
Yeah.
You know, and then
You learn
so-
to hate these people.
We all love our coaches.
Actually, I mean, so Lee is actually even understating how important this is. I think, believe it or not, he can understate this. I mean, so all my friends on Facebook know I lost the 50 pounds, right? But when I go to my own doctor at Sutter Health, which is neither Stanford or UCSF, it's another group, and it's in the newspaper, they spent $1 billion on their electronic health record, Epic. That my doctor was the last one to know that I lost 50 pounds because their $1 billion system doesn't talk to my $40 gadget. I mean, can you smell the opportunities here? It's unbelievable. But you know, so we in medicine, I know there are a lot of medical types in the audience, we in medicine have got to get with the program that the patients, especially the well ones, will start to get advice from these types of systems, para-health types of systems, electronic systems, because our doctors are going to be the last ones to even know what's going on in their lives. It's a big problem for us. So.
Get them to start answering their email. How about that?
Yeah, the email's another one.
Next question.
Hi, I'm kind of surprised this hasn't come up, but the issue of access to these medicines and money. And so Jimmy Carter was in this trial. He's an ex-president, so he probably has some access. I'm guessing, is this a clinical trial that he's in, or is it?
No,
Okay.
actually, it's an approved drug.
Okay, but I'm guessing that if I got metastatic melanoma, that I might not have access to the same drugs, or I may not be able to afford that. And with personalized medicine, it seems like it becomes even more expensive. So I'm just wondering what the panel thinks about what the possibilities of getting this out to everyone who might need this drug, as opposed to just those who have access.
It's a very good question.
It's a fantastic
and it's
question.
about, what's the price on this? About $150,000?
Yeah, it's very expensive. And but we do everything that we can, and we do a lot to make the drug available to everyone who needs it. Fortunately, metastatic malignant melanoma is not a common disease. And so it is possible for us to do that through a variety of assistance programs. But the reality is that these advances that we're talking about, at least when they start at the beginning, are very expensive. And how you get those things out to the general population is very important. One of the things that we did early on when we recognized that we had a drug that could do good in this way was to create an expanded access program which would make it available to anyone, both in the United States and in Europe, who met the qualifications of having the disease in which we'd shown that the drug was efficacious. And we continue to do that as we broaden the set of possible places where the drug can be used. But the question you raise is a really important one, which is how does society actually pay for these kinds of advances? As it happens, most of those individuals who develop advanced malignancies are, a majority are in an elderly population. And so we're really talking about the United States government and how the United States government pays for this, because people are Medicare eligible in most cases. And that's an important thing for us as a society to decide what's worth paying for.
The only thing that I would add to that is that scientifically, I would say the rationale would point to even more and more expensive drugs. Because if you take the example of, say, Merck's Keytruda, it works by releasing a brake on the immune system. And one of the interesting ideas is, can you combine other drugs with that and even do better in terms of getting a higher rate of remissions among more and more patients? And so you might sort of release the brakes and put something else in there that throws some fuel on the fire to really get the immune system to go after that tumor. And that's a second drug. And that's probably going to cost a lot too. So,
Well, our view of that is a little bit different. I mean, we have by far and away the largest number of combination studies being done, but we are specifically doing this with the idea in mind that we would hold the price constant. Our goal is to reduce the price every year. And so, our sense is that as combination therapies can be made more effective, there's a broader set of the population that could actually use them, and we could actually reduce the price. Of course, you're talking to the R&D guy here, you're not talking to the salesperson, so we'll see where that gets to, but that's certainly our view in the laboratory.
Yeah, I guess also I would like to introduce and remind people that everything we just talked about here kind of affects maybe the United States and Europe and Canada. But in fact, there are billions of people out there who literally you can't get an anemia diagnostic to. And so I think we need to ask the questions as we think about these things, which is, it's almost like the arguments that go on around what's happening to the economy or otherwise, which is, do you create a bigger divide? And how do we make sure that maybe the tools that we're using for our personal, you've got one on here, monitoring, are there ways we also think about leveraging the innovations that are happening there to help us bring along the portion of the world that's been left behind in terms of their health? Now, obviously, $150,000 drugs are not going to be happening for people who are in the poorest populations. I'll just tell you that, for instance, in Malawi, a very poor country of 6 or 8 million people, they basically spend $31 a year on health care on average per person. So we're not talking about anything near here. So I think it's important for people to also keep sort of a realistic view about perhaps there's opportunities to leverage these innovations and ensure that they transition in a way that isn't going to necessarily take a $150,000 drug and turn it into a 30-cent drug, but in fact will take the learning and turn it into something that can be more broadly available. So.
Yeah, that's, an example of that is some years ago, we identified a drug that could be used to treat a variety of worm infestations in animals. We were able to use that to develop a treatment for river blindness, onchocerciasis. And we made the commitment as a company that we would provide that drug to everyone who needed it for as long as they needed it. We have given out now more than 2 billion doses of that through the Mectizan donation program. And already we have 3 countries in the world that are free of this disease. And we think we may be able to, for the first time, eradicate this disease around the world through yearly administration of this drug, Mectizan, which also works in another rare disease called elephantiasis. So we have the ability to take discoveries that were really hard won, very expensive to generate, and to make them available for underserved populations around the world. And we've done that not just for Mectizan, but for our vaccine programs, particularly in India, and a whole variety of others to improve the health care of people around the world.
Roger, let's give you a hard time here for a second, though, okay? So GSK announced over the weekend.
Please do.
GSK, GlaxoSmithKline, announced over the weekend that they're not even going to file patents on their molecules now in the developing countries.
Right, which is a little bit sleeves off your vest.
Yeah.
Right, because they just do them anyway, so it doesn't
well,
matter.
And to pick on Roger's former company, Amgen, it's finding out how difficult it is to sell a high-priced medicine in the United States, even with really compelling biology. Has anybody heard of the PCSK9 cholesterol-lowering drugs? These are about twice as effective as statins at lowering LDL cholesterol. It's really exciting biology, really good clinical trial data. They came out and priced these things at $14,000 to $15,000 a patient, and the payers across the United States have said no.
I mean, so
Not prove it that you can reduce heart attack and stroke, and not until then.
But instead of picking on the pharmaceutical companies and biotechs, which do great work, for a second, I think probably Roger would also agree. So in my view, with precision medicine, which we've been talking about, there are going to be so many diseases and sub-diseases and subcategories. There are not enough pharma and biotech companies on the planet, actually, to help us all, even if they're all 100% successful. I mean, we need a different way to think about it. And so what I try to do is just try to get young kids, college students, graduate students to think about what's a more efficient way to develop drugs. Because I don't think it's sustainable the way we can do this. We're not going to get the drugs in our lifetime for all of these diseases we need. I think we just got to get more people thinking about it.
Well,
Okay,
but
we
I mean,
got more
keep in
questions, and I
mind,
wanna-
keep in mind that the cost of drugs is a very, very small part of medical care. And it is less than 10% of the cost of medical care and has been less than 10% since the time that effective therapeutics first became available, antibiotics. The fact is that we should all recognize there is no country on earth that can afford its healthcare system, period.
that is
There is none. And so this is a problem for the entire world.
Question on the end.
So this is also for Dr. Perlmutter. There's a work of a mycologist by the name of Paul Stamets who's local, and there's some sort of semi-famous anecdote about him curing his mother's stage 4 breast cancer with some extract from turkey tail mushrooms. And it seems to be connected to this unlocking of the innate abilities of the immune system. Do you know of any connection of that? Do you know of any correlation of this sort of mechanism there that might be similar to what your drug does?
You know, I
Any
don't.
connection there?
I don't.
There are clinical studies of this drug, of this mushroom extract that are in clinical studies now.
Yeah, there are a variety of natural products that have been studied, you know, to look to see whether or not they have activity either in a preclinical setting in an animal model or in looking at tumor cells in a dish, or in some have made it into clinical trials. In some cases, that we've obtained important drugs that way. It does happen, but it is very much the exception.
What piqued my interest was that the
often
mode
not.
of mechanism he described in his TED Talk sounded very much like what your drug did.
Right, and that's been as I say, that's been a popular mechanism. There was these were more than 100 years ago. There were things called Coley's toxins, which were thought to work by stimulating the immune system to reject tumors. They might actually have worked that way. It was basically stimulating the immune response. We're not sure, but in today's environment, we have a lot of much more sophisticated tools that we can use to identify how these things work.
Question here on the side. Yeah, you've been patient.
Oh, hello, my name is Anustra and I'm a junior at Interlake High School. So Dr. Butte, you mentioned something a while ago about wearable technology, and this reminded me of a study I read a while ago about microchip, implantable microchips. And I was wondering what, like from the perspective of those microchips being used for a medical perspective, I was wondering what the panel thought about this type of wearable technology from the perspective of either data sciences or from personalized medicine.
Yeah, so first of all, isn't it awesome there's a high school kid here?
Yeah.
There needs to be more of these, so obviously we gotta get more of them here. So for implantables, I think, I don't know, maybe Lee knows more, maybe you guys know more. I think you can get to obviously a lot more of the measurements like the blood with implantables than the external stuff, though you can get to a lot of blood stuff too. So diabetics now, some diabetic patients wear continuous glucose monitoring, things that are external but with catheters inside. I don't know. I mean, I think that there are patients that wear internal types of chips. I think it's a research study, but it's just amazing how many consumers are buying the external stuff, right? Even without a doctor convincing them to do it, or even insurance covering it, right? I mean, people buy these things. So I think there's just a lot of growth in that kind of area. I brought a lot of props, so now that gives me a cue to bring up one of my props here, right? So talk about inventions here for a second. So what's really cool to me too is how easy it is to make these types of devices. So a medical device like that would be implantable, or in let's say 5-10 years ago, a medical device might take a $50 or $100 million investment to get a company launched. So there's a company called Scanadu. Any of you heard of this? Raise your hands if you bought one. There's one hand, two hands there. So Scanadu, about 3 years ago, said, well, let's make a medical device. This is what it looks like. This is it. That measures your pulse oximetry and your blood pressure, your heart rate variation. And they launched this on Indiegogo. Okay, you know what Indiegogo is? Like Kickstarter. And they wanted to raise $100,000 over the weekend to make this medical device. They had raised $1 million over the weekend, right? To launch this thing, because all of us prepaid $200 to be a pre-customer. It's like, we don't care when you send it, but we want one of these because it looks like a tricorder from Star Trek. That's how they kind of launched this thing, right? And it kind of like glows red and stuff like that. I mean, you can see it's like charged and stuff. So this is what it takes to make a medical device now. You don't even need like a bazillion dollars. You could actually launch one of these with a good team, and you're full of good teams here in Seattle. With just Indiegogo and Kickstarter. I mean, it's actually possible to do this. What an amazing time this is, right?
Oh, okay. Also, Dr. Hood, what would you consider this sort of wearable technology for projects such as the 100K Wellness Project or just personalized medicine?
I want to turn that over to, what wearables are you guys using at Arrowvale and all these
So
others?
we're using the simplest. We're using Fitbits, and I'll say they do two really important things, three really important things. They can keep track of altitude gain, they can keep track of steps, and they kind of keep track of the quality of sleep. So those are three fundamental things. I'll tell you myself, I think there are a lot of other more complex variables that I think are really critical. And by a complex variable, I mean a variable that integrates together the behavior of two interesting systems. And Atul mentioned one, heart rate variability integrates together the parasympathetic and the sympathetic nervous systems. It's enormously informative about the quality of your health.
That's right.
So Microsoft is putting out an equivalent to the Fitbit, which they call the Band, and they can beautifully do heart rate variability plus steps plus all the other things. So I think we're in an evolutionary stage. We ought to keep tuned, but I would guess within 6 months, Arrowvale will switch from Fitbit to something that'll take us up to the next level.
Yeah, I mean, so these are pretty sensitive. I'll just give another story here. So in my part of the country, we have earthquakes, right, in Silicon Valley. And so
we do here too.
Yeah, I know, but yours make news. Ours are like wine bottles break, but which is a tragedy in itself. But so sometimes these happen in the middle of the night, and so I think Jawbone did this and Fitbit did this, where so many people use these now that they can see a peak of everyone turning over in their bed with an earthquake. So the last one we had, I looked myself. I could see. I woke up. I turned over in bed with the. You can mash that up with USGS kind of data. That's how sensitive these things are. I mean, they're not perfect, but don't shoot for perfection. It can even tell there's an earthquake on our way.
Almost as good as dogs barking.
Yeah, that's right. They couldn't tell me a minute in advance, but told me there's something going on here. So that's the solution. I think it's external ones. I don't know how many people voluntarily put the stuff in them because the external ones are so good now. So.
Thank you.
Okay, next question.
So you had previously talked about immunotherapy. I was wondering how these new immunotherapy drugs for cancer could possibly be kind of engineered in a way to help cure or treat autoimmune diseases.
Yeah, great question. So if we can do something to rev up the immune system, can we work on the other side and turn it down? And there's a huge amount of interest in that. Over the last 20 years, there's been really quite a lot of progress in making drugs to treat autoimmune disease. But we still have a ways to go, and this opens up a potentially important avenue. It's probably worthwhile my saying, unsurprisingly, that if you have a drug like pembrolizumab or Keytruda, the one that we make, or nivolumab, Opdivo that Bristol Myers makes, which are the two drugs that have been registered for cancer that rev up the immune system, that these drugs do cause autoimmune disease in some percentage of patients for exactly that reason. As you turn up the immune system and you get responses to your own joints and skin and parts of your intestinal tract, it's not so good. But that helps to tell us also that there is hope that maybe we could use a similar kind of thing to be able to turn it down. We do have to recognize that there's the possibility that if we turn it down too much, that cancers that previously were being held in check could emerge. So we have to be a little bit careful about it. It takes a lot of study.
Well, and there's opportunistic infections. If your immune system is...
Well, always, but the current therapies have that problem too.
Do you think we understand the immune system as much as we understand cancer? I mean, we've had a war on cancer. We seem to know a lot about cancer, but immune system and immunity seems like it's still far behind. What do you think?
Well, I mean, I think the reality is that we don't understand enough about either.
Yeah.
The problem at this point, so that put us all in the same place, we in the biopharmaceutical industry are pretty good at what we call composition of matter, at making new materials that have never before been seen on Earth and that can do something specific, that, you can take a pill and it will actually get through your intestinal wall and make it into your blood and go where it's supposed to go and hit what it's supposed to hit. We just don't know what we're supposed to make it to do because we don't really understand how the machine works. We don't understand human physiology well enough to know what we're trying to fix. I mean, the real problem is if you don't know how the machine works, then it's pretty hard to tell what's wrong with it when it's broken. And then you don't know what to fix. And so this is a plea for more basic research because that's what we really need. If we had a much better understanding of diseases like asthma and rheumatoid arthritis, we'd know how to intervene, but we don't because we don't understand it well enough.
And that's the industry guy making the argument for basic research.
Absolutely.
That's very well said. That's great. Yeah, please, next question.
Quick one. Cancer tagging, cancer cell tagging, is that does it lose currency with the immune system intervention, or is that just as effective, or is there a new generation of it?
Tagging it to find out where the cancer cells are, or tagging it to get rid of it?
Well, both.
Yeah. I mean, I think we're always interested in any new approach that helps us identify cells that are bad actors and make them do something else, either kill them or change their behavior in some way. We're very excited right now, as I've said, about the fact that we have a systemic therapy that seems to take advantage of the ability of the immune system to kind of do that work for us. But just as Luke mentioned, it is a minority of patients for most cancers. For melanoma, it's actually the majority of patients we can get responses. But people with lung cancer, for example, it's only about a quarter or so. And so that means 3/4 don't get responses. And the question is, how do you get responses in those people? And improved ways of identifying which are the bad acting cells, the malignant cells, and targeting them in such a way that we can get better immune intervention. That's something we clearly want to try and do.
Thank you.
Okay, we got one more coming up here on the side.
Hi. My son had a very rare disease that's now cured. And there was a lot of basic science research out there that no medical system accessed. I had to.
Yeah.
I'm wondering about dense data clouds in terms of using fundamental findings that are out there to make things, to make get clinical results from the basic science that's out there.
Well, I'll tell you, I think Iceland is absolutely the ideal place to use these dense, dynamic, personalized data clouds. Number one, they have a genealogy that starts at 900 AD, and almost all of 300,000 people who live on the island can map themselves into that tree. And what that means is, number two, a company called Decode has generated an enormous amount of sequence information, 10 or 15,000 complete genomes and many partial genomes. And from that information, you can begin to make inferences about the nature of the genome sequences of individuals who've never had their genome sequenced. And what I think would be ideal would be to add to that genetic information the environmental information that comes from clinical chemistries and proteins and metabolites in your gut microbiome. I think that would be an absolutely ideal experimental place to begin solving really fundamental problems, since we can make so many predictions about their genetics, and we now have the ability to measure much of their environmental triggers. So it's a great place, and we're gently exploring the possibility. Wouldn't it be exciting if we could have a whole country move toward the kind of wellness I've talked about tonight? That would be utterly transformational for the country.
I think this is one of the really big opportunities, and one of the things that we're seeing come up is both basic research and clinical science engage more with citizen science. There's a a patient group that I interact with, the Grace Wilsey Foundation, which is it's an incredible story. You should Google this if you haven't seen it.
I'll tell the story if you're not going to.
So I'll tell a little bit of it. So there's a guy, and his daughter had some pretty bad symptoms, and he couldn't figure out what was going on, and they couldn't diagnose it. And so what he did was he went on Google and he took out AdWords for her symptoms. And anytime any other parent would Google for these symptoms, they would get to the webpage. And so he amassed a cohort of patients, and they started sequencing them, right? The way Atul was talking about sequencing individual people. And they figured out the mutation. It's a mutation in a single gene called NGLY1 that causes this disease, right? This is entirely driven by the patients. And so this is something that I think clinically and, on the basic research level, we need to be open to this, right? We need to encourage people to do this. I mean, obviously, patients are the most driven to be healthy. And we need to do more to reach out.
That's absolutely right. So I was part of the team that found that NGLY1 mutation for the Wilsey Foundation. So first of all, when you start a comment like my kid had a disease, that can go one of two ways. So I'm glad that actually medicine worked out here. But you're highlighting the point I think the entire audience has to get. You care about your own health more than anyone else, okay? I know the doctors, we do care, but you care about more than anyone. And that means you got to own everything that we can give you. That means owning the data. So it only worked with the Wilsey Foundation because we gave the genome results back to the patients, right, to the family. They say, here's the PowerPoint we just showed you. Here are the genes. And you start Googling up to see what they mean because this is as far as we know how to go. The doctors, we care a lot, but we are way behind, and you cannot, as a doctor, you can't always have faith that we're going to know everything. I mean, that's just a fact of life. We're going to have to depend more and more on computer systems to tell us what to do. Or if the right patient comes in and the right medication's available, the decision support system in that complicated system is going to have to tell us what to do because medicine's gotten very complicated, as any doctor would tell you. So what I would say is that you are going to have to own your medical system, your own medical care. You're going to have to want to learn how to download data. You're going to learn how to start to get second opinions and things. But we have to, in medicine, empower you to really get all the data you need. It is not simple. I mean, if your own doctor doesn't know what to do, you're on a search again. You might have the name of a gene or a condition, but it's a long search to figure out who the experts are to get to a treatment. And I'm glad it worked out in your case.
I'd add one thing to that. I think saying you should own your data is wrong.
Okay.
My own feeling is society has created the tools that let us do these wonderful things. And we all have an obligation to give back to society our de-anonymized data for inventing the medicine that's going to take care of your kids and your grandkids and things like that.
Yeah, and actually, I agree 100% with Leanne. When I say own, I mean you want to be responsible for it. We are going to need your data, but you're going to need your data too. Data is a kind of weird thing where you can have it and I can have it. It's not like a car where I'm borrowing it. It's not like water. We are finicky with water in California. If I drink it, you can't have it, right? But data, you can have it and I can have it too. We can do amazing things, both of us.
So you ought to have access to it.
Yeah, that's exactly right.
And we need better tools for your access so that you can take your data and understand what it means. Yeah. I mean, most people just don't have a chance now, and we can change that.
This is really a great place to wrap up that intersection between science and society. And kudos to all of our panelists for coming out here and talking about what they do. Can everybody please give them a round of applause? So I know Lee wants to spend a couple of minutes on some closing remarks and talk about the ISB symposium.
I think
Maybe
we're
just a
all
minute.
ready to go home. So I would say I hope you get a sense of the fact we're at many different tipping points now. Many things are changing, and for all of those who have young kids at home, tell them this is the most exciting time ever to go into science. I mean, what they can do is going to be unbelievable with just the kind of tools we've heard talked about here. Tomorrow we'll have the second day of a symposia called emerging technologies and systems biology. And this morning we went through just a wonderful, marvelous set of discussions. And tomorrow, actually, if you really want to hear the latest in immunotherapy, you can come and hear Roger give a keynote talk first thing in the morning on immunotherapy.
Or you can sleep in.
But there are many other fascinating talks. There is room. If any of you are interested, come by and you can join us. And I'd like to extend my thanks too. I think it is wonderful to see this many people come to this kind of panel discussion and be focused and to have the kind of questions we got tonight, which were superb, and from young people as well as older people. So thank you very much.
All right.